Can you people see the screen? Okay. Am I audible and can you people see the screen?
Yes, ma'am.
Okay, fine. Now we have to see some food tests. So we say that there are different tests which can be used to detect the presence of starch. We have reducing sugars. Then we have proteins and fats and oils. And then we can see one test for vitamin C as well. Okay. First, we have to see the test for starch. Now, in case of the detection of starch, what you need to do, you have to add drops of iodine solution to the food sample. Now, if the food sample contains starch, so the color of the, you can say the color will change from orange brown to blue black. So that means that shows the indication of starch, right? That shows the presence of starch in the food if the color changes from orange brown to blue black color. Is that clear? And normally this experiment is done on potatoes, right? Potatoes do contain starch, so you can see the visible change in the color of potato, like the place where you are going to drop the iodine solution. Then comes the test for reducing sugars. In this case, you have to add Benedict solution into a test tube that contains the food sample. Now, then you have to heat it. You have to heat at 60 to 70 degrees centigrade in a water bath for 5 minutes. And again, you have to observe the color change. You have to observe the color change of the solution. So if the reducing sugars, you can say if the reducing sugars are present in the food sample, in that case, the solution will turn the solution will turn from blue to orange or brick red color. So the color change for reducing sugars is from blue to orange or brick red color. So that indicates the presence of reducing sugar in the food sample. Next comes the test for proteins. In this case, you have to add a few drops of you have to add a few drops of Biuret solution to the food sample, right? To the food sample solution. So if there's protein present, in the solution, the color change will be from blue to violet or purple. So in the test for proteins shows the color changes from blue to violet or purple. Then comes another test that is done for fats and oils.
And is it also known as the ethanol emulsification test?
Yes, fats and oils. In this case, you have to add you add 2 centimeter cube of ethanol to a test tube with food with the food sample, and you have to shake it well. Then you have to pour the ethanol into an equal volume of cold distilled water. Now, if the substance contains, because if the substance contain,
Fats, then the test tube will become cloudy.
yes, very good. If the test substance contains lipids, so you're going to see a cloudy emulsion will form that indicates the presence of lipids. Now we have to see the test for vitamin C. In this case, you have to add 1 centimeter cube of DCPIP solution to a test tube. Okay. What is this DCPIP solution? Yes, students. Yes, any one of you knows about it or not? Ma'am Sam, do you know about it?
No, ma'am, I only know that it's used for the test of evidence.
Okay, it's actually called dichlorophenolandophenol. It's quite big name, right? And it's used for the detection of vitamin C that is also called as ascorbic acid. So you have to add 1 centimeter cube of DCPIP solution to the test tube. Then you have to add some food sample solution. Now, if the food sample contains vitamin C, then the blue color dye blue color of the dye of DCPIP will disappear if the vitamin C is present in it. So the blue color of the dye, it changes or disappears. So that is how you can detect the vitamin C presence in the food sample. Is that clear? So these were, I guess, 5 practicals that were related to this chapter named as biological molecules. Yes, ma'am, Sam, have we covered all the practical? Okay. Which of the following orders would be correct showing the size of structures from the biggest to smallest? What do you think? I'll ask you
C.
one. Okay. According to you, that is C. That was Mipsam.
Yes, ma'am.
Okay, that is correct. I'll ask you one by one, and then you have to give me the answer. This is just for your practice. In this question, yes, Ganya, this question is for you. The diagram below shows part of an organism's ciliated epithelium. What term would correctly describe the level of organization of the structure in the diagram above? You can make a guess, Ganya, it's okay. I'll tell you the right answer. Okay. Uh, Laiba, are you there? Laiba, could you hear me? Yes, Laiba, am I audible to you? Okay. So the diagram below shows part of an organism that is ciliated. Okay. Mipsam, could you guess the answer for this question?
It's a cell.
Very good, Mipsam. Okay. So in question number 6, they ask you about which structures are present in plant cells but not in animal cells. Yes, Mipsam.
Ram, cell wall, chloroplast, and sap vacuole.
So which option would be the best one according to this?
I guess B.
B, yes, so you are absolutely correct. Laiba, can you hear me? Okay. Mipsam, could you tell me the question numbers like the ones you were talking about? Are they related to the hard questions?
Ma'am, the last ones, I mean, they were theory questions. Yeah.
Question.
No, no, not this one. Uh, can you scroll down a little more? They were about keys. Was it this one sheet or was it the
Okay,
other
this
one
is
about
related to the
keys?
chapter 1. The keys one
Okay,
must be
okay,
from chapter 2.
okay,
Okay, so
okay.
we can solve these ones as well. We will do that later in the upcoming class. So the image shows a palisade mesophyll cell found in the leaves of plants. Which row of the table below correctly shows the structure labeled on the cell in the image above? Uh, you need to see both of them. Yes, Mapsam, I believe that only you are in the class right now. So, yeah.
Ma'am, why is the
Okay,
vacuole
just wait a minute, Mapsam. Laiba said so that I don't know. I have seen her message. Okay, Laiba, you can answer yes. Let Mipsam answer this question, then you will answer the later questions, right? Okay, Mipsam.
Ma'am, X is the mitochondria, Y is the vacuole, and then uh, and then no, X is the chloroplast. Yeah, X is the chloroplast, and then Y is the vacuole. So then the
Correct
option
option
is C.
C.
I
Yes.
said C.
Okay, that's correct. Good. Okay. Yes, Laiba, you can try this question. A student cut a section through a plant, finding a group of long microscopic structures which lacked end walls or any dry or any cell contents. Which row of the table below correctly identifies the structure described in the statement above? I have to see. Laiba. Okay, Laiba, you have to answer this question. Okay, Laiba said so that the answer would be D. Yes, Raiba. The answer is that hollow lignified tubes to allow uninterrupted flow of water. Good, Laiba. You're absolutely correct. Okay. This question is for MEPSA. A diagram of an animal cell. Identify the parts labeled A, B, and C. State the names of two structures in plant cells that are absent in animal cells. State the name of one structure that is present in bacterial cells and in plant cells but absent in animal cells. So yeah, start with the first one, Mipsam. Identify labels at A, B, and C.
B is the cell membrane, C
Okay.
is the nucleus, and then B is the sorry A is the cytoplasm. Um, yeah, B is cell membrane, C is the nucleus, and then A
Okay.
is the
Cell
cytoplasm.
membrane. A is cytoplasm and C is nucleus.
So
Okay.
in
This
two,
yes.
ma'am, yeah,
Okay. Now next question is for Laiba. I think she has replied. Uh, Chloroplast and central vacuum. Let me see. State the names of two structures in plant cells that are absent in animal cells. Okay. The first answer was she said chloroplast and central vacuole and cell wall as well. Yes. We can take cell wall, uh, and chloroplast. Then state the name of one structure that is present in bacterial and plant cells but absent in animal cells. Yes, Mibsam.
cell wall, cell.
Yeah, bacterial cells and absent in. Okay. Okay. Then comes shows a bacterial cell. Identify the structures labeled A to F. Yes, Laiba, could you label these structures? see A1, A, B, C, D, E, and F. Okay, A is cell wall. I'm going to label it here. Cell wall. What about B? B is cytoplasm. No. You see that lab here they have mentioned this. They have labeled this B. So it's actually the ribosome. It's ribosome, right? What about C? Uh, okay. Uh, the rest of this, Mipsam, can you tell the rest
Yeah,
from
ma'am.
C?
C is the cell membrane.
Good.
Uh, then D is the cytoplasm.
Okay. That, yeah, that's good. E's,
And is E the genetic material?
yeah, but it does have a name. That is plasmids, right? The circular genetic material, the circular DNAs, right?
Yes.
What about F?
F is the circular DNA. Uh,
Yeah, actually, F is the, yeah, we can characterize F as a circular DNAs. Okay, now yes, how the circular DNAs and plasmids differ. Yes, Mepsa.
yes, ma'am.
Like, what is the difference between these circular DNAs and plasmid? Actually, okay, it's better that we revise it, right? The circular
Yeah,
DNAs
ma'am.
are, okay,
Yes.
the circular
Uh,
DNAs are, you can say these are the main genetic material of the bacterial cell, right? It is the one large circular molecule of DNA. While the plasmids are, you can say they are small, circular pieces, pieces of DNA, not the whole DNA, right? They are circular pieces of DNA that are separate from the main chromosome. And you know that a bacterial cell can have one, many, or none, no of the plasmids. And also, the plasmids, you can say that they carry extra, or you can say non essential genes, right? Those genes could be antibiotic resistance genes, genes for toxin production, and then genes for conjugation as well. While the circular DNA It floats freely in the cytoplasm, and yeah, the circular DNA contains the essential genes, right? Now, those essential genes could be either for growth, metabolism, then comes reproduction as well, right? Is that clear? So this one's the circular DNA, right? And these are the plasmids, the smaller ones. Is that clear, Laiba and Mepsam?
Yes, ma'am.
Okay. So yes,